Okay. So we'll continue from where we stopped in the previous lesson. We have looked at the structure of the mitochondrion, right? So we will continue with the function of the mitochondrion. Now over here, this is a three-dimensional structure of a mitochondrion that is cut into half. I want you to look inside the mitochondrion. Can you see the nail-like structure there? All over the entire inner membrane of mitochondrion, right? You can see all the nail-like structure like this one here, right? Okay, so this is a crista, which is an inner membrane that protrudes into the matrix of the mitochondrion. Can you see this structure here, which looks like a nail? So this structure is called stalked particle. Okay, stalked particle. Now this stalked particle is actually looks like a nail, right? Like this. Now the part that looks like the head of the nail contain the enzyme that is necessary for the synthesis of ATP. So this enzyme will catalyze the synthesis of ATP. That's why this is a site of the respiratory ATP synthesis. So this is where ATP will be synthesized in the mitochondrion. Okay, so now my question to you is the ATP being synthesized in A, crista, B, inner membrane of mitochondrion? Which answer is more accurate? A or B? Please type your answer. A or B. So the answer is B. Why is B more accurate than A? You see, if your answer is A, this means you're telling me that the crista is the location where ATP is synthesized. That means you will have missed out the non-crista part, which is actually the inner membrane of mitochondrion. Okay? Inner membrane is more accurate because crista is also inner membrane. It's also made up of inner membrane. So if you say inner membrane, you are correct. If you say crista, you are half correct, half wrong. Half correct, half wrong in A level is 0 mark. Okay? Your answer that's half correct, half wrong in A level is 0 mark. So you want to achieve total correct answers. Okay? So this is how a mitochondrion looks like in the cell. You can see the finger-like projection, the cristae inside this organelle, right? Usually it's sausage shape. So if this is an animal cell, are you able to recognize the mitochondria inside this cell? Look for organelle that's slightly sausage shape and there are some finger-like projection inside it. Okay. Of course, the big one here is the nucleus. Now, can you see this sausage shape? And then got some finger-like projection inside it, which is not clear, but you can actually see them, right? Something like this extending outwards. Now, if you can see this one, for the same tone, you can see the rest of them because it's slightly grayish patch, like this one here, right? This one, this one, this one, okay, this one, all these are mitochondria. This one might be mitochondria, but I'm not sure because not very clear. Right? If you can identify one, you can identify the rest of them. So I can give you a micrograph in exam, ask you to, okay, label 2 mitochondria, okay? Or better still, label the organelle that synthesize ATP, then name them. So that means you label and name them mitochondria because I asked you to label 2, right? Okay. Okay, let's look at the function of mitochondrion. Now, if you've got the notes and you've got a PowerPoint, if there are differences between the two, the most accurate one is my PowerPoint because PowerPoint is easier for me to modify than the notes. The notes is PDF, it's more difficult for me to modify, okay? So therefore, the one in my PowerPoint will be more accurate, okay? If you've got 2 different versions. All right, function of the mitochondrion is the site of ATP synthesis. And it is synthesized by the process called aerobic respiration. Aerobic means you need air. Air means oxygen. You need oxygen. Okay. Now, don't forget ATP can also be synthesized in the chloroplast. You will learn this one in your A2. All right. So what is the role of ATP in the cells? It is a universal energy carrier. Okay. Why is it called universal? Now, when you say if you travel a lot, you will be bringing universal adapter, right? That means no matter which country you go to, regardless of what is the shape of the plug point, you will be able to use your universal adapter, right? And then you can charge your handphone, you can charge your iPad, and so on. So universal here means apply in everywhere, but apply where? So It is universal, means applied in all the cells in our body and all the cells in all organisms, you name it, plant, fish, animal, okay? So universal. What about energy carrier? That means it carries energy. ATP is a molecule that carries energy. Now, in biology, when you say it carries, you must tell me carry from where to where. Okay, carry from where to where? Carry from energy yielding reactions to energy requiring reactions inside the cell. So the cell carry out a lot of reactions. So the ATP will carry energy from those energy yielding reactions to those energy requiring reactions inside the cell. So it's like you taking your handphone, you go and charge your handphone, right, at the plug point there. So that the plug point function is to release the electric current for you. So energy yielding reactions. And then you will bring your handphone around whenever you need to do your job. Okay. So you go to the energy requiring reactions. Okay. So the ATP is like the battery inside your handphone. It's a rechargeable battery. So it will allow your handphone to carry out energy requiring reactions. Okay. Right. So imagine this box is a cell, right? Inside the cell got many reactions. We divide them into energy yielding reactions and energy requiring reactions. So energy yielding reactions, when they yield means release the energy will be used to make ATP from ADP and PI. So this process is called condensation. Okay, when PI is added to ADP. Now, the meaning of PI, the I here stands for inorganic. P means phosphate. Okay, inorganic phosphate looks like this. So PO4 negative is an inorganic phosphate. Okay, why is it inorganic? Because there's no carbon and hydrogen inside. That's why it's inorganic. Okay, ADP plus PI becomes ATP. So this one needs energy from here. This process is called condensation. It's also called phosphorylation because phosphor means you're adding the phosphate to the ADP. Okay, so after ATP is formed, ATP is a very small and soluble and mobile molecule. Mobile means can move. So it is an intermediate molecule, means in between. In between what? In between energy yielding reactions and energy requiring reactions in the cell. So it will carry energy between energy yielding reactions and energy requiring reactions in the cell. Okay, so it moves and then it will then release its energy. Right? So how does it release the energy? It will be hydrolyzed. So ATP hydrolyzed to ADP and PI, and 30.5 kilojoule of energy released per mole of ATP hydrolyzed. So this energy will be used by the cell to carry out, to do certain work inside the cell. So what are the work that the cell needs the ATP to do is anabolic reactions, active transport, and movement. So these three main work will need ATP inside the cell. Okay, so the cell trades or donate ATP, meaning, I want to introduce you to the term energy currency. ATP is an energy currency of the cell. You know, currency means money, right? Currency means money. So you imagine the cell is Malaysia. Anywhere you go to Malaysia, you want to buy something, you want to get some services, you need to pay with the currency ringgit Malaysia. So same thing, you go to anywhere in the cell, you ask that part of the cell, hey, organelle ribosome, I want you to synthesize protein, can or not? Can. Pay me with ATP first. Okay? Or you go to memory and say, hey, I want you to carry active transport for me, cannot? Can. Pay me with ATP first. So therefore, ATP is the energy currency of the cell. Okay? So the cell will have to trade or to donate or to pay ATP in order to ask the cell, certain part of the cell, to do the work. Okay? So these are the three types of works that will require ATP in the cell. First is anabolic reactions. Now, you know that all biochemical reactions in the cell. Now, biochemical reactions, all the reactions in the cell which you can write in the form of chemical equation. So all these processes are called metabolism, right? So all biochemical processes in the cell is called metabolism. And metabolism, you learned already that it can be divided into anabolism as well as catabolism, right? Anabolism is a building up process. Okay, hang on. So anabolism is a building up process, whereas catabolism is a breaking down process. Okay, so let us look at the building up process. It is the building up process that requires energy. So what are they? I'm going to erase this away. Okay. So anabolic reaction is a building up process, or another word, synthesis, right? Because you take simple molecules, you join them up, they become complex compounds, right? So for example, in photosynthesis, CO2 and water are simpler molecules, and you join them up to form the more complex molecule like carbohydrate, okay, sugar. So this is a anabolism. Respiration, when you activate the glucose, you add another molecule to the glucose. This is called anabolic reaction as well. So it need ATP. The rest of them are the synthesis of molecules in the cell, like DNA synthesis, when you join nucleotide to form DNA, protein synthesis, when you join amino acids to form polypeptide. Okay, glycogenesis is the synthesis of glycogen. So you add glucose together to form glycogen. Starch synthesis, you add glucose to form starch. Lipid synthesis, maybe you convert glucose to triglycerides, or you join 3 fatty acids and 1 glycerol together. So all these are synthesis part under anabolic reactions, all will require ATP. Okay. Now the next one is active transport. Example, sodium potassium pump. Sodium potassium pump, in order for them to pump sodium and potassium, we need to trade ATP to them. Okay. Last one is movement and locomotion. Any movement in the cell requires ATP, right? For example, muscle contraction is due to the movement between actin and myosin. So this movement needs ATP. Movement of cilia and flagella, when they beat, because their movement, you also need ATP. Endocytosis and exocytosis is also movement of the cell. You imagine, this is a cell, and it wants to carry out phagocytosis, right? Endocytosis, for example, phagocytosis. So first, let's say you've got a bacterium here. So it wants to engulf the bacterium. So in order to engulf the bacterium, can you see that the cytoplasm, right, must stretch outwards towards this bacteria, isn't it? It must stretch outwards, right, forming the pseudopodium. So the stretching outwards, right, you can see that there is a movement of the membrane to move towards this direction. So this movement also need ATP1. Okay, so this is phagocytosis. Exocytosis, you got this phagolysosome, right? Move and fuse with the membrane to release undigested material. So this is also a movement, it needs ATP. Lastly, movement of vesicles on microtubules. Now, any organelles that move inside the cell, they don't just move freely. They have to move on a track, just like your LRT, right? Your LRT, they have to move on the track. So they got the monorail track. So same thing, any organelles that move inside the cell, they have to move on the monorail, all right, the track. So your movement of transport vesicles to Golgi apparatus, from Golgi apparatus to Golgi vesicles, all will move on a monorail, okay, a track. So all these movements needs ATP. Chloroplasts, remember chloroplasts, they can move inside the cell. So the movement of chloroplasts also, they're moving along a track, okay? So the role of ATP in the cells, in short, is for the cell to do their work, such as anabolic reactions, active transport, and movement or locomotion, all right? So the next organelle we want to look at is microtubule. The word micro stands for small or tiny. Tubule, when you got a bule there, means it's a little. Tube stands for pipe. So pipe is hollow one, right? Okay, a hollow pipe. Okay, inside got space one. So that's called the pipe, the tube. So what is microtubule? Microtubule is a long rigid hollow tube. Do you know the meaning of the word rigid? Rigid means not easily bent and break. So you cannot bend them, not easy to bend. So long, rigid hollow tube. Diameter is 25 nanometer. So this is similar to the size of the ribosome, right? Okay, the size of the ribosome for eukaryote. Now, what is it made up of? It is made up of protein subunits called tubulin. So each of these is a tubulin molecule, which is a protein molecule. Okay. Now, we have got two types of tubulin, the alpha tubulin and the beta tubulin. So that's why the color is different. It means they are slightly different. So when alpha and beta tubulin, they join together, they will form a tubulin dimer. Okay, tubulin dimer. Dimer means two together, two join in one. All right. Now, then the tubulin dimer will join end to end. So now here you got one tubulin dimer, right? So you got another tubulin dimer will join here. So that means the end of this tubulin will join to the end of another tubulin. The end of this will join to the end of the other. So they join end to end. So these both sides are both ends. These both ends will join to both the ends of the other tubulin. So they join end to end. So this is joining end to end like this here. Can you see? Joining end to end. One end joined to the other end. So when they join up like this, they will form a long strand like this. So this long strand is called the protofilament. So it's like a strand, like a string, right? You know, like a string. You put all the necklace together in one line, right? So this is a string, and your necklace got pink color and white color. So string of pink and white alternate color of pearls, right? So we call this protofilament. Okay. Okay, so tubulin dimers join end to end to form the protofilament. All right, what is protofilament? Now, proto stands for first. Have you heard of the word prototype? Prototype is the first sample that you have created, right? Proto, first. Filament comes from the word fili. Fili stands for thread, means it looks like a string. So protofilament is the first string being produced when the tubulin dimer joins end to end. All right. So once they join end to end, they form a tubular structure where one ring is made up of 13 protofilaments. Can you see this one ring? So this one ring is made up of the 13 protofilaments. So this whole thing is called the microtubule. All right. So microtubule is found inside the cytoplasm of the cell. There are 4 functions for microtubule: A, cytoskeleton; B, anchorage; C, monorail; D, building blocks of centriole cilia and flagella. So let's look at the function one by one. Okay, first one, it is a cytoskeleton. Cyto stands for cell. Skeleton stands for framework. Okay, framework. Framework means something that supports the body. For example, we've got skeleton, right, to provide the support to our body. So the cell also will got that internal skeleton, internal framework, and the internal framework is made up of microtubule. Right, so we call the internal framework the cytoskeleton. All right. So the cytoskeleton is a complex network of interlinking 3 types of molecules: microtubules, intermediate filaments, and microfilaments. So the one that we want to learn here is only microtubules. Okay, so microtubules will form part of the cytoskeleton of the cell. So can you see in the diagram on the top right hand corner, you look at the tubes inside the cell, right? There are 3 different colors and sizes. The biggest one is microtubule because it's tube-like, okay? The rest of 2 of them are filaments. Filaments are thread-like, so they are smaller. So they're all interconnected to form a network of the cytoskeleton. So the function is to give shape and support to the cell. So you know that cells got their own shape, like red blood cell is always biconcave this shape. So there must be something inside there to maintain and support this shape. So that is a cytoskeleton. And then sperm cells always this shape, right? Okay. And then our nerve cell is this shape. Okay. So why is it the cell got their own shape? It's because there is there is cytoskeleton to provide it the shape and support. Okay. And these cytoskeletons are continuously being disassembled, reassembled, and rearranged. The reason is because if a cell there need to consistently change shape, then the cytoskeleton will also have to consistently being assembled and disassembled. Okay, say for example, this is, let's say, your white blood cell that phagocytoses, and then suddenly this bacteria is here. And your white blood cell wants to go and engulf the bacteria, right? So originally already got the microtubules here, say for example, or interlinked network. But it wants to extend the cytoplasm out to engulf this bacterium. Then you have to push the cytoplasm out, right, using the microtubules, okay, as well as the intermediate filaments and microfilaments. So therefore, these microtubules are consistently being disassembled and reassembled and rearranged in order for the cell to consistently change shape to carry its function or for it to move around, right? So this changing of shape is due to, for the process of exocytosis, right? Something moves towards the membrane and fuse with it, okay? Endocytosis. Mitosis is because you use the microtubule to form the spindle fiber, so you need that. Cell movement, which is amoebic movement of the white blood cell. So white blood cell, right, moving and it wants to move here, you have to build more microtubules here. And then it moves here, you have to break the microtubules here. Right? So consistently being assembled and disassembled and rearranged. Second function is anchorage. Okay. Anchorage is you want to fix the organelle in certain location of the cell, just like you want to fix the ship in a certain spot in the sea, you don't want it to move. So you put an anchor into the sea. So here you put an anchor, you use a microtubule to anchor the organelle in a certain location of the cell. So for example, here you can see that this mitochondrion is being anchored in its position. The Golgi, the rough ER also is being anchored, right? That function is monorail. So we have got microtubule. So this one is the microtubule here. Can you see this diagram? This is a microtubule. You can see the tubulin dimer, right? You've got two different colors there, right? Tubulin dimers. Okay. So microtubule forms the monorail for the organelles to move. Can you see this organelle, which is pink in color? And can you see this one, which looks like the leg? So you start to move along the microtubule, right? Like the movement of the transport vesicles, right? So all of them will move along the microtubule. A movement of Golgi vesicles or movement of secretory vesicles, all are moving on the microtubule. So microtubule act as monorail. Lastly, building block of centriole, cilia, and flagella. That means centriole, cilia, and flagella, all these three are made up of microtubules in it. In it, yes, okay. So we'll study these three structures now. First one is a centriole. Now, when you see that the word equals to, that means it means the definition of. Centriole is equals to what means centriole's definition is. It is one of two, because centriole always occurs in pair, right? So centriole, when you see the word centriole, it means one. So one of the two small cylindrical structures, so cylindrical, okay, made up of microtubules. So this cylinder, this whole thing is made up of microtubules found in the centrosome near the nucleus. So if this is a cell and this is the nucleus, you will find the centriole in a region near to the nucleus. So this region here is called the centrosome. Okay. Now, this centriole is made up of 9 triplets of microtubules. So what do you mean by 9 triplets? You see here, this is a triplet of 3 microtubules fused together. So it's called a triplet. So in a centriole, it's made up of 9 triplets. So that means they arrange in a circle. 9 triplets arranged in a circle. Right? So microtubule is made of 9 triplets of microtubule. No, sorry. Centriole is made up of 9 triplets arranged in a circle. And the length is 500 nanometer. Diameter is 200 nanometer for the centriole. Okay. And centrioles usually appear as a pair, always 90 degrees to each other. So like this one here, they are 90 degrees to each other, or like this is 90 degrees to each other. Okay, like this is 90 degrees to each other, always 90 degrees. Okay, can you see these two? They are 90 degrees to each other because one is this orientation, the other one is this orientation. So always 90 degrees. So what is the function of centriole in the cell? They form the poles during mitosis and meiosis. How to memorize? You see centriole ends with the word O-L-E-O. The pole also ends with the alphabet O-L-E. So centriole is used to form the poles during mitosis and meiosis. Okay, so can you see this pole here? During mitosis, this is mitosis. This is actually metaphase of mitosis. Can you see the pole? You can only see one here, right? Okay, and you see one here, all right? This is dark colors because all the spindle fibers are all attached and accumulated there, so it looks dark, but it's actually not centriole. Only this one is a centriole and this one is a centriole, right? So the pair of centrioles separate during mitosis to form the pole, each one of them. So in each side of the pole, you only can find one centriole, right? The centriole also form the bases of the cilia and flagella. So those bases are called the basal bodies. Bodies means structure, something that you can see, something that you can see. Basal means at the base of, okay? At the base of something is called basal. Okay. Now, centriole was believed to act as MTOC. Last time, we thought that centriole is MTOC. MTOC stands for Microtubule Organizing Center. So based on this term, you know that this is a place which organizes the microtubule. That means this is a place where microtubule is being synthesized and they're being arranged to form centriole, to form cilia, to form flagella. So we thought centriole got this function, but actually it's not the centriole that carries this function. It is a centrosome that acts as MTOC. All right. So what is centrosome? Centrosome is an organelle. Now, usually when you come across the word some, right? Some stands for body or structure or sometimes organelle, right? Some means it's a structure that you can see. So centrosome, central means in the center, that means near the nucleus. So it's an organelle found in plant and animal cells that is situated near the center of the cell, that means near the nucleus. All right, so it's outside the nucleus, near to it. Okay. So the function of the centrosome is to synthesize the microtubules, and then after that, they will assemble the microtubules into centriole, cilia, and flagella, and also spindle fibers during mitosis and meiosis, and also cytoskeleton of the cell. Okay, now we're going to look at the structure cilia. Okay, cilia. Now, cilia, the word is for plural, singular is cilium. Just like one bacterium, many bacteria, right? One bacterium, many bacteria. One cilium, many cilia. Okay? Definition, it is a whip-like structure. You know the whip that you use to beat something, okay? Rotan, okay? Whip-like structure projecting from the surface of some animal cells and the cells of unicellular organisms. That means if this is a cell, it is projecting outwards from the cell, right? Projecting outwards from the cell. So this is the cilia. So let us study the structure of the cilia. Now here what you can see is that this is a membrane of the cell. Can you see or not? Okay. And this is a projection projecting outwards from the cell, right? It's still surrounded by cell surface membrane, but it's projecting outwards. So this is called the cilium, one cilium. At the base of the cilium, where you can find the centriole, it is called the basal body. So centriole function here is for the anchorage of the microtubules from the cilium. All right. So basal body is replicated from the centriole. So it's formed from the centriole, the basal body. So this is the basal body here. Okay. So how do they look like? Basal body actually looks like this, and cilium inside actually looks like this. Can you see the arrangement of microtubules in both of them are different? One is 9 triplets arrangement, the basal body. Hang on. And the other one is 9+2 arrangement. Okay, let us study the 9+2 arrangement of the microtubules in the cilium. Now, the 2 stands for 2 singlet microtubules. Right in the center of the cilia, you've got 2 individual microtubules that are not joined to each other. So we call them 2 singlet microtubules for these 2 here. Then the 9 is because of the microtubules that are arranged at the side. They appear in doublets. So you've got A microtubule and B microtubule. They join together to form a microtubule doublet. So microtubule doublet, okay? They're joined together. So altogether, we got 9 of them, 9 of this MTD, microtubule doublet, at the outer ring of the cilia, all right? That forms the outer ring of the cilia. So therefore, it's 9+2 arrangement of the microtubules. And then the A microtubule has got 2 arms, the outer arm, which is this one here, and the inner arm. So it looks like this. This is microtubule A, this is microtubule B, and this is the arm. So if this is the outer part of the cilia, this is the outer arm, and this is the inner arm. Okay, so this is A and this is B. So imagine A is somebody that is driving the motorbike. B is the passenger behind A. Okay? So the outer arm and inner arm is made up of dynein protein arm. So it's made up of dynein protein. So the protein is called dynein. So the arm is made up of dynein protein. Okay? So here in the cilium, we can divide it in 2 parts. The cell surface membrane, which is the blue color one, and all the microtubules in them, they are all named together as exonym. So the entire structure and arrangement there is called the exonym. So cilium is made up of cell surface membrane and exonym. So exonym is the cylindrical structure inside the cell surface membrane, which is made up of microtubules. So this is the structure of the cilia. Okay, microtubule, we already learned there are 13 protofilaments, but so this is for A microtubule. Now B microtubule is incomplete because it attaches to A microtubule. So can you see the B microtubule? It is an incomplete ring because it attaches to A. So it is only made up of 10 protofilaments instead of 13, the normal one. The complete one is 13, but this one is only 10. All right. So this is a comparison between, okay, hang on. Okay, we always talk about cilia and flagella. So now we want to compare between cilia and flagella, okay? Now, cilia and flagella, interestingly, they've got the same definition. Okay, both of them got the same definition. So, wheat-like structures projecting from the surface of some animal cells and the cells of many unicellular organisms. So, definitions is the same. And both of them are made up of 600 different types of polypeptides. Just now, you only learned maybe 1 or 2 types only. Actually, there's 600 different types of polypeptides in order to allow fine control of the beating movement of cilia and flagella. Okay. Now, comparing between cilium and flagellum, one cilium, many cilia. one flagellum, many flagella. All right. Cilium is shorter versus flagellum is longer. There will be many cilia per cell, but only one or two flagellum per cell. The beating mechanism for cilia and flagella are the same. The beating is due to the dynein arms. Remember, this is A microtubule and B microtubule. All right, B microtubule. A microtubule got two arms, right? So these two arms will go and attach to the B microtubule of another doublets. And then because this is a filament, right? So you move up and down this tube. So as you move up and down this tube, then the cilia and flagella will also start to move. Okay, so this is how the cilia and flagella move. The beating mechanism is due to dynein protein arms coming into contact and then move along the neighboring B microtubules. So that is the moving mechanism of the cilia and flagella. Okay, so I'm going to show you exactly how they move afterwards. Cilium is by beating or rowing movement, whereas flagellum is wave-like movement. I'll show you afterwards. So the function of cilium is, if the cell is not attached to something, it is to move the cell. But if the cell is attached to something, it cannot move. The movement of cilia is to create a sweeping current for something to be moved, something inside the fluid to be moved. For example, dust particles, bacteria, viruses, all trapped in the mucus in your lungs, right? In trachea, bronchus, bronchiole. You use the cilia to create the current to sweep the mucus so that bacteria, fungus, viruses will be swept out of your airways, all right? And also, you know, the egg, once it's released to the oviduct, so the cells of the oviduct will contain cilia to create the current for the mucus to move so that the eggs will be swept along with the current of the mucus. Okay, so this is the movement. Then for flagellum is to move the cell. So to move the cell means locomotion, movement of organisms. Okay. Now, what's the difference of the movement between cilium and flagellum? Cilium will move more effectively in one direction. So let's say to the left side is power stroke. Okay, it's like you're swimming, right? Power stroke. So you are actually pushing your arm to the back inside the swimming pool in the water. So you need power to do that. So this is called power stroke. Then recovery stroke is when you bring your arm back to the front, you don't need much energy. So the pushing to one direction is stronger than going back that direction. So that means you're creating a current that moves things towards this direction one. Okay, more effectively towards one direction. So that is for cilium. So it's similar to the arm of a swimmer doing the breaststroke. Flagellum movement is wave-like, so it moves this way like wave. Okay. Now this is example of cilia and flagella found in the cell. First one is paramecium. Paramecium, can you see surrounding the paramecium got a lot of hair-like structure, so they are the cilia. So it's to help in locomotion of paramecium. Whereas for ciliated epithelial cells in our body, they cannot move. So they're attached to our connective tissue. So what moves is actually the cilia to move to create the current of the mucus so things can be transported together in one direction. Okay. For flagella, it's usually for the movement of the cell, for example, sperm cells and euglena. Okay, that's all for today. We will stop here today.